This topic is divided in two parts, and this corresponds to part 1, and this will have focus on prebiotic and evolutionary perspectives on nucleotides and biosynthesis and regulation of purine nucleotides. The life is configured to evolve with an outstanding properties of nucleotides. Metabolic requirement of earlier form of life met with a versatile functioning or a multifunctional abilities of nucleotides. Nucleotide as a monomer serve the function of chemical energy, carrier of chemical energy. Energy quantum which is released is utilized, is coupled to make unfavorable reaction favorable in metabolism. In second place, nucleotide as a discrete monomer evolved with certain modifications such as an attachment of amino acid, an attachment of vitamins, an attachment of subhydryl groups, and they oriented in a way to do redox chemistry and a group transfer chemistry in metabolism. Alongside these reactive nucleotides could have assisted to co-evolve RNA polymers around them to bear a function to act as a catalyst, RNA enzyme, or ribozyme, such as ribosomes. In a third place, polymers of RNA or polymers of nucleotides that randomly or catalytically assemble together and bear the property of replication, and that transform everything and serve the basis to evolve in a new way. As we know, the nucleotide called ATP and GTP, they are carriers of chemical energy, and they are working since then till now. As we know, the compounds which act as coenzymes like nicotinamide adenine dinucleotide, flavin adenine dinucleotide, S-adenosyl methionine and coenzyme A, they act as a coenzyme and they are working since then till now. Some of the functional assemblies of RNA molecules bear the functions of catalytic RNA, and we know them as ribozymes and ribosomes, and they are working since then till now, as we know, ribosome, which is a crucial part of protein synthesis. Overall, the polymers acquired ability of replication through autocatalytic property or by means of other assembled RNA molecules which assisted them to have that property of replication, and that rose as a sense of information, storage of information and transmission of information. Overall, all these properties of nucleotides seems to be RNA-centric, and RNA that played a pivotal role in a hypothetical RNA world, and alongside it also served a basis to new way to evolve. No other molecule other than nucleotide in the history of world of non-living things and world of living things could have evolved with an outstanding, unique property, and that property laid a new dimension to chemical evolution of life. And that unique property is intermolecular complementarity. This strand serves as a template and invites other complementary nucleotides to have a pairing through hydrogen bonding, and that's how the complementary base pairing takes place. And this strand, in some sense, provides some sort of information that act as some information which is stored in that strand, and that strand is copied as a complementary strand, and that complementary strand, when further copied, that gives exact original template, DNA template, and that is how it provides some sense of storage of information. It provides some sense of information storage and transmission through complementation, and that complementary strands serve as an intermediate between two sense strands that having exact information lies within, and that has led a way for Darwinian evolution through selection and evolution through selection, and that led the RNA to evolve to DNA and the DNA protein world that came into existence onwards. This is an overview of RNA and DNA with its canonical building blocks. As you see here, adenine and guanines are derivatives of purines, and cytosine, uracil, thymines are derivatives of pyrimidine. When these nucleic bases are attached on a ribose sugar, the constitution, the assembly is called nucleoside, and when the nucleoside is further attached with phosphate group, that assembly is called a nucleotide, and that nucleotide form a linkage through 3' to 5' phosphodiester linkage, and that's how you get a polymer of RNA and polymer of DNA. DNA you have deoxyribose sugar, RNA you have ribose sugar. Let's have some prebiotic evolutionary perspectives on nucleotides. Much before life came into existence, there were prebiotic chemistry which plausibly synthesized nucleotides. Nucleic bases such as the purines and pyrimidines could have synthesized utilizing prebiotic chemistry, utilizing prebiotic conditions of primitive earth, and a plausible synthesis of purines and pyrimidine nucleotides. In 1960, John Uro, a Spanish biochemist, along with his team, was able to show the synthesis of purines and pyrimidines by utilizing primitive conditions, prebiotic conditions, and hydrogen cyanide. A polymerization of hydrogen cyanide could give an intermediate which is actually present in a biosynthetic pathway, and that intermediate could have further modified with hydrogen cyanide, cyanate, and cyanogen to give various canonical and non-canonical nucleic bases such as guanine, such as adenine, guanine, hypoxanthine, xanthine, and so on. Similarly, polymers of hydrogen cyanide gave cyanoacetylene and cyanoacetaldehyde. Fusion of cyanoacetylene with cyanate and fusion of cyanoacetaldehyde with urea could synthesize cytosine, and similarly other pyrimidines could have been synthesized utilizing the same chemistry, and this is a condition, a plausible condition could have gave rise to various nucleotides much before the life came into existence. And here we go with some structures of hydrogen cyanides. These are a fusion of hydrogen cyanide with another hydrocyanide that gives a dimer that is further fused to have a trimer and then to have a tetramer. And this tetramer can further treated with Pyrimidine, or it can exposed to sunlight, and that gives an intermediate called aminoimidazole carbonatide that is further hydrolyzed to get an intermediate of purine biosynthetic pathway called aminoimidazole carboxamide. And this intermediate can be further modified with hydrogen cyanide, cyanate, cyanogen, and that can provide various bases synthesized such as adenine, hypoxanthine, xanthine, and guanine. Similarly, you have hydrogen cyanide that is termed as cyanoacetylene, which is further treated with cyanide, and that can give you cytosine. Similarly, cyanoacetaldehyde can be fused with urea and you get a cytosine synthesis done. This is a ribose sugar when fused with triphosphate that gives you carbon 1 to cyclic phosphate, and when this cyclic phosphate is coupled with various nucleobases such as adenine, cytosine, uracil, guanine, and that gives you another intermediate called ribonucleotide to phosphate. Further modifications of ribonucleotide to phosphates give you ribonucleotides, and that's how the polymer of RNA could have synthesized in prebiotic conditions. This is coenzyme A, nicotinamide adenine dinucleotide, and this is 3-phosphoadenosine phosphosulfate. Notably, all these have adenine monophosphate as a common scaffold on which certain modifications have made. In case of NAD, this is a nicotinamide which is acted as a reactive group to have a reversible redox chemistry. In case of coenzyme A, you have a sulfhydryl group that acts as a donor of acyl and acceptor of acyl and do redox chemistry through thioester bond, and you have 3-adenosine phosphosulfate, and here you have sulfate group attached to phosphosulfonhydride bond. This is more similar like phosphoanhydride bond which is present in ATP, and similarly it transfers this group, a sulfate group, the way ATP transfers phosphate group by cleavage of phosphoanhydride group at gamma position perhaps. Notably, all these molecules are ancient molecules. They are working since earlier form of life, still a current form of life. They have been conserved, and they could have assisted other RNA assemblies to evolve around them to have some catalytic activities, and they bear a function such as RNA catalyst, RNA enzymes, ribosomes. And this is ribosome. This is the large subunit of ribosome. Notably, you can see a blue portion which is RNA portion, and a green portion which is protein portion. The red dot shows the catalytic site of large subunit of ribosome, and that is deeper inside in ribosomal RNA. Ribosomal RNA bears the function of catalyst, and the protein which is present around is serve the basis of providing structural stability to the ribosomal proteins, and altogether that acts in a protein synthesis. So in the first place, a ribosomal RNA that acts as a catalyst and do peptidyl transferase activity in ribosome. That is called, in other words, it is called peptidyl transferase ribozyme. Later on, through evolution, RNA enzymes gradually replaced with protein enzymes, and protein enzymes came into place to into a current form of life. Here you can see RNA that can be converted into deoxyribonucleotide through an enzyme called ribonucleotide reductase. And this enzyme, evolution of this enzyme turned to be instrumental in switch from RNA to DNA in an evolutionary process. So RNA is evolved to DNA through the activity of ribonucleotide reductase enzyme that seem to be pivotal in evolution from RNA to DNA world. And this is a extra structure of ribonucleotide reductase class 1 enzyme, and there are three classes of ribonucleotide reductase enzymes which do a redox chemistry and turns ribonucleotides into deoxyribonucleotides, and that is evolution from RNA to DNA protein world. Let's switch to protein biosynthesis and regulation. Purine biosynthesis takes place through a couple of pathways, primarily that is de novo pathway and salvage pathway. De novo pathway begins with metabolic precursors such as amino acids, ribose phosphate sugar, which participate in this biosynthetic process, and that is being taken from pentose phosphate pathway. Then you have bicarbonate, ammonia, which all together work for purine biosynthesis. A salvage pathway which recycles the prebases and nucleotides released from nucleic acid metabolism. This is a picture that shows scientists G. Robert Greenberg and John Buchanan, whose team in 1950 pioneered the work of purine biosynthesis along with their team, and a meticulous work for 10 years discovered all the steps involved in purine biosynthesis. Origin of ring atoms of purines comes from radioisotope experiments performed by these groups by using carbon 14, nitrogen 15 labeled precursors. This is a list of enzymes which are involved in purine biosynthesis. They are, in a way they are participating into purine biosynthesis. They are put as a list. The first enzyme is initiate a committed step of purine biosynthesis, and this also acts as a regulatory step of purine biosynthesis. The enzyme is glutamine phosphoribosyl pyrophosphate aminotransferase. Second enzyme is glycinamide ribotide synthetase. Third one is glycinamide ribotide transformylase. Next is formyl glycinamide synthetase. Next is aminoimidazole ribotide synthetase. Next is aminoimidazole ribotide carboxylase. Next, succinyl aminoimidazole carboxamide ribotide synthetase. Next one is adenosyl succinate lyase. Next one is aminoimidazole carboxamide ribotide transformylase. And the final enzyme of purine biosynthesis is cyclohydrolase. Their short forms or abbreviations are put in parentheses. And this is a list of products that are formed in a sequence. The letters in bolds are abbreviations of the products called phosphoribosyl pyrophosphate, and in parentheses the name of enzyme being included. Second product is phosphoribosylamine. Next is glycinamide ribotide. Next one is formyl glycinamide ribotide. Formyl glycinamide ribotide. Aminoimidazole ribotide. Carboxyaminoimidazole ribotide. Succinyl aminoimidazole carboxamide ribotide. Aminoimidazole carboxamide ribotide. From aminoimidazole carboxamide ribotide. And the final product is inosine monophosphate. This is an overview of purine biosynthesis. Phosphoribosyl pyrophosphate modified sugar with pyrophosphate group participate into a biosynthesis pathway. Glycine participate into a first step of purine biosynthesis pathway and displaces it's a glutamine that participate into first step of a biosynthesis pathway and displaces the pyrophosphate, and you get beta anomeric configuration of sugar, and that onwards act as a scaffold to synthesis of nucleobase called hypoxanthine. In the first step, glutamine that donate amide nitrogen on a carbon 1. The next step that forms an intermediate called phosphoribosylamine, and in the next step, glycine participates, and it's a ATP driven reaction where you get intermediate called glycinamide ribotide. In the next reaction, N10 formyl tetrahydrofolate participate into reaction, and that donate formyl group and an intermediate which is formed that is called formyl glycinamide ribotide. In the next step, second glutamine participate into the reaction. It's an ATP driven reaction, and that contributes to an intermediate called formyl glycinamide ribotide. In the next step, imidazole ring is formed, and that gets closed through ATP driven reaction mediated by aminoimidazole ribotide synthetase, and the intermediate that formed is called aminoimidazole ribotide. In the next step, bicarbonate that participate into the reaction and provides 6 carbon to the growing purine ring, and the intermediate which is formed called carboxyaminoimidazole ribotide. In the next step, aspartate that participate into the reaction, into a biosynthetic pathway, and the intermediate which is formed is called succinyl aminoimidazole carboxamide ribotide. Next to it, the carbon skeleton of aspartate is being removed as a fumarate, and an intermediate which is formed is called aminoimidazole carboxamide ribotide. In a consecutive reaction, another N10 formyl tetrahydrofolate participate and provides another formyl group to the growing chain, and that acquires carbon 2 position of a growing ring, and the intermediate which is formed is called formyl aminoimidazole carboxamide ribotide. The final step is closure of the ring through condensation and elimination of a water molecule, and that gives an nucleobase synthesized, and that nucleobase is called hypoxanthine. Notably, none of the constituent nucleotide is synthesized, but it's a non-canonical nucleotide, a nucleobase which is synthesized as a ribonucleotide called inosine monophosphate, and that nucleotide serve as a precursor for further synthesis of adenosine monophosphate and guanosine monophosphate. Let's switch to structural depiction of purine biosynthesis pathway. Here you have a ribose phosphate which comes all the way from pentose phosphate pathway, and that is further modified with it's an ATP driven reaction that donates pyrophosphate group, and the modified sugar is called phosphoribosyl pyrophosphate, and that participate into purine biosynthesis pathway. A first step, which is mediated by glutamine phosphoribosyl pyrophosphate aminotransferase, act as a committed step of purine biosynthesis. Similarly, it also acts as a regulatory step of purine biosynthesis. Glutamine, which participate in a initial committed step, and that provides amide nitrogen to take an initiative for purine synthesis, and that displaces this phosphate group and acquire a position of nitrogen 9 on sugar, and that is a beta anomeric configuration where the sugar plane, the amide group, amino group, which lies above the plane of sugar, and rest of the purine biosynthesis assembly of purine base takes place in beta anomeric configuration. In the next reaction, glycine participates into the reaction, and this is the ATP driven step where carboxyl group of glycine form an carbamide linkage with amino group on carbon 9, and that acquires carbon 4, carbon 5 and nitrogen 7 position on a growing purine ring, and that forms the intermediate called glycinamide ribotide. In the next reaction, N10 formyl tetrahydrofolate participate and donates formyl group, and it's a free amino group is attached with formyl group, and that gives an intermediate called formyl glycinamide ribotide that acquires carbon 8 position of growing purine ring. In the next step, second glutamine participate into the reaction, and it's also ATP driven reaction where it acquires nitrogen 3 position of growing purine ring and an intermediate which is called formyl glycinamidine ribotide. In the next step, the ring is closed through ATP driven step, and you get imidazole ring synthesized, and the intermediate is called imidazole ribotide. The next step, carboxyl group from bicarbonate participate into reaction, and that acquires carbon 6 position of growing purine ring. An intermediate which is formed is called carboxyimidazole ribotide. In the next step, aspartate participate, and amino group of aspartate is attached to the carboxyl group, a free keto group of growing purine ring, and that's form a carbamide linkage. In the next reaction, that forms an intermediate called succinyl aminoimidazole carboxamide ribotide, and this linkage is cleaved in the next reaction. The carbon skeleton of aspartate is cleaved as a fumarate, and that is released, and you get an intermediate called aminoimidazole carboxamide ribotide. In the next reaction, a second N10 formyl tetrahydrofolate participate to this pathway and donates another formyl group at second position. It requires second carbon position of bromine ring, and in a final step the ring gets closed. Before that, an intermediate which is formed with the formyl group that is called formyl aminoimidazole carboxamide ribotide, and in the final step the ring is closed and you get nucleobase called hypoxanthine done, and then nucleotide which is called inosine monophosphate. This is a non-canonical nucleotide which is synthesized in a purine biosynthesis pathway that can be taken further to synthesize constituent nucleotides called adenine monophosphate and guanine monophosphate. Similarly, there is no participation of free ribose sugar in early stage, but it's a phosphate ribose that participate into the pathway and provide ribose sugar which act as a foundation or scaffold on which purine ring is biosynthesized, and you get inosine monophosphate synthesized that act as a precursor for adenosine monophosphate and guanosine monophosphate. Inosine monophosphate can taken further to synthesize adenosine monophosphate and guanosine monophosphate. In a couple of steps, in the first step of adenosine monophosphate synthesis, enzyme called adenylosuccinate synthetase, and this is X-ray structure of an enzyme, various enzymes involved in adenosine monophosphate synthesis and guanine monophosphate synthesis. In the first step of adenosine monophosphate synthesis, an enzyme called adenylosuccinate participates. It's a GTP-driven step where aspartate is contains with hypoxanthine ring, and that provides amino group to the hypoxanthine ring, and the carbon skeleton of aspartate is in the next step is removed as a fumarate, which is quite similar a step involved in a eighth step in a purine biosynthesis, where fumarate is released and an intermediate called succinyl aminoimidazole carboxamide ribotide is synthesized. So, in a step of adenosine monophosphate synthesis, the condensation of aspartate gives an intermediate called adenylosuccinate, and that is further gives adenosine monophosphate through cleavage of fumarate, and this is an amino group attached on the sixth position of purine hypoxanthine ring, and you get a nucleotide done called adenosine monophosphate. In the case of synthesis of guanosine monophosphate, it's again a two-step reaction where you have enzyme called inosine monophosphate dehydrogenase. It's a redox enzyme that oxidize hypoxanthine ring, and it's a dehydrogenation of oxygen at 2 position, and NAD as a coenzyme is being utilized, and that is being reduced to NADH, and an intermediate which is formed called xanthosine monophosphate. This is further amidated with the help of glutamine. The amide group of glutamine is being utilized to provide an amino group at second position in a synthesis of guanosine, and you get guanosine monophosphate synthesized. An enzyme that acts for this step is guanosine monophosphate synthetase, and this is notably an ATP-driven step that turns xanthosine monophosphate into guanosine monophosphate. Let's switch to the regulation of purine biosynthesis pathway. The initial two steps of purine biosynthesis are mediated by ribose phosphate pyrophosphokinases, and a next step, which is a comminuted and regulatory step, which is mediated by glutamine phosphoribosyl aminotransferase. Guanosine monophosphate that can be turned into guanosine diphosphate and guanosine triphosphate, and similarly adenosine monophosphate can be turned into adenosine diphosphate and adenosine triphosphate by utilizing various nucleoside phosphatases, kinases, nucleoside kinases that acts on GMP and that provides phosphate group, and they are turned into guanosine diphosphate and consequently guanosine triphosphate. So we have synthesis of GTP, and similarly we have synthesis of ATP, and all these di and triphosphate along with monophosphate nucleotide inhibit or regulate the activity of ribose phosphate pyrophosphokinases in first place, and then phosphoribosyl pyrophosphate aminotransferase is being inhibited by guanosine monophosphate, guanosine diphosphate, GTP. They binds on allosteric region of glutamine phosphoribosyl pyrophosphate aminotransferase. Similarly, adenosine monophosphate, adenosine diphosphate, and ATP can bind to another regulatory site of glutamine aminotransferase, and they regulate the synthesis of IMP, that is inosine monophosphate, is regulated through a regulatory action exerted by guanosine monophosphate, diphosphate, triphosphates of guanosine and adenosine. The next level regulation takes place at this branch point, where IMP is used as a precursor to synthesize guanosine monophosphate and adenosine monophosphate. Guanosine monophosphate and adenosine monophosphate acts as a competitive inhibitors of IMP, and consequently they act as regulators of their own synthesis. So the synthesis of GMP and AMP is kept in a check, that there is no more inappropriate synthesis of IMP that is leading to nucleotide synthesis. On the other hand, GTP and ATP coordinate in a reciprocate manner on synthesis of guanosine monophosphate and adenosine monophosphate. ATP regulates or coordinates the synthesis of guanosine monophosphate, whereas GTP coordinates the synthesis of adenosine monophosphate. They act on enzyme involved in GTP synthase, that is GMP synthase, and enzyme involved in AMP synthesis, that is adenosine succinylase. So that's a coordinated regulation of guanosine monophosphate and adenosine monophosphate synthesis, and that ensure that the coordination ensures that there is a appropriate amount of GTP and ATP to take it further for nucleic acid biosynthesis. And that's all for this. Thank you and happy learning.
That is it.